So as previously, welcome questions from anyone online in the room. Please introduce yourself. I suppose I should invite, first of all, questions of fact for David, if anyone has one. Yes.
Yes, thanks. Very interesting, David. Just one quick question. I thought I understood from Becky's presentation that for these practical trials, you have to assume that the relative treatment effects are the same in every randomization list. And that would clearly not be the case because of these site-specific, species-specific effects that you mentioned, David. Have I got that wrong?
You won't be surprised to answer. I'm going to give that to somebody else.
So yes, it is a big assumption to make, and it's an assumption that we looked at in our simulation study. We tested the network meta-analysis approach in situations where there were differences across randomization lists. We pushed it quite far, and it does still do very well, even when the relative treatment effects are different across sites. But it is something we will also test in the analysis, and we could, if necessary, produce rankings for different sites.
Thank you. I guess you could sort of stratify the analysis somehow by species distribution or something like that. Just a thought. Yeah.
So, I mean, effectively, I mean, the approach, part of the approach is a stratification approach. And, I mean, I think, yeah, absolutely, Richard, we plan a lot of... We've thought about these issues, we plan a lot of analysis. There is a degree to which you've got to run the trial and then we're going to work it out a bit as we go along. And I think it does raise a broader question, actually, about whether you think it's more important to randomise and reduce systematic bias, even if you don't have all the answers, or, you know, kind of say, actually, this is too hard a problem, so we just won't randomise and we'll, you know, go with... I mean, you know, philosophically, it's an interesting question.
Other questions?
Oh.
Good. Question over there?
Hello, Holly Jackson from Geneva University. I have so many questions, but maybe I'll just stick with two. Sarah, you mentioned, and we discussed earlier, about the fact you can't really have a type 1 error in these types of trials. Is there any, I don't know, backlash from that? Are there discussions that you've had where people don't like this because there's no type 1 error, and how did you deal with that?
So I think it's a really good question, and coming back to the point I made earlier about clinical context, Holly, I'm not suggesting this is an appropriate design for regulatory or licensing. So this is really a design in the situation where you've got lots of things that are being used, could be being used, and you've got no evidence. And I think in that context, at the moment, I haven't had a lot of pushback because to a degree, the kind of idea behind a type 1 error is we know this works and we really, really don't want to make a mistake and say something is better when it isn't. But actually, when you don't know anything, yeah, it's a bit of an artificial dichotomy. And I think, coming back to, Max didn't really talk about it this morning very much, but if you think about the whole MAMS design, there's a recognition there that there are lots of new things and we don't know which is better, and therefore we have a complex design. And I would argue that in the way we've talked about practical, there is a recognition that there's this morass of things that we could give, and we've got no way of identifying at all what we might do. And therefore, you don't get the pushback on type 1 error. I don't know, Michael, anyone else wants to?
I'd just like to extend the question, actually, which is how do you know when you get your results that they aren't just noise?
Well, you rely on the simulations, which tell you that 1,000 patients gives you, 90% of the best you can do. I mean, Ian, I do think, the concept of ranking is very powerful. And it does come back to this issue that actually, if there's one thing that is really terrible and the ranking can confidently exclude that, is that good enough? Because I still think the ranking is more likely to be unbiased than what you get at the moment, which is, real world evidence, which we all know is deeply systematically biased. So I think you're actually coming back to my original point, which is about how do we value systematic versus random bias in situations where we don't have a lot of information. I don't know. Do you want to answer your own question?
Not really.
Sorry, could I just
Holly,
ask?
you had another question.
If that's okay. You in the last presentation you talked about adding and dropping arms. How do you do that with this design? I would love to know.
Becky?
So we're going to look at probabilities that an arm would be the worst. That's right, isn't it? We're going to have a criteria, and if there is a certain probability that an arm is the worst, then it will get dropped. That's right, isn't it?
And we're also, I mean, as David said, we've also thought quite hard about single arm monitoring, which we're increasingly using actually. So this concept that actually some things may just never get over the bar to make it into those, you know, if you're an anti-venom which causes, 25% of people to have a severe anaphylactic shock, then no one's going to use you, whatever you do, efficacy-wise.
I'm going to take a question online now. This, I think, is one for you, David. How would informed consent work in an anti-venom trial? People suffering from snake bites may be required to consent in a medical emergency. How does it work practically?
Yes, so we've adopted a very standard approach, I think, now for sick patients effectively, in which we assume consent. If there's no relative, if there's a relative there, we get consent from a relative or a guardian, and then as soon as the individual is well enough to actually sign consent, we go back and get consent. If at that point they refuse, then we don't use any further data from on there. I think it's pretty well established in head injury trials, etc., that it's now become standard.
Okay, thank you.
Can I just add?
Oh, sorry.
Just to sort of add on that, I think the key thing, of course, is that you're very much in the practical design where actually you're engaging the centre in the decisions around what sort of kind of what medicines are appropriate for them. So it's actually quite a different sort of kind of trial design in the sense that you're not going there and offering them the protocol and saying, here is the protocol. It's saying, well, here is the options of the different regimens. And, can you consider what would be appropriate for which babies for these particular sort of syndrome or clinical infection type, if you like. So therefore, what we're identifying so far is the buy-in from the unit is therefore much more. Obviously, the standardization of the issues of deferred consent or others are standardized. But I think the key component to that is actually from the clinician's perspective, a part of that, they're very engaged in that process, I think, to do that. Yeah, thanks.
Thank you. Take the question from Simon and then on the end there.
One way of defining statisticians is people are obsessed with conveying uncertainty, like standard errors, confidence intervals. So if you're using ranks, any good or bad practices there? You mentioned putting a probability on the chance of it being worst or best. And from a clinical context, do you know or any thoughts about how having uncertainty about rank would change how the ranking is interpreted, i.e. if it's possible to change the treatment based on for each individual patient as they turn up? I don't know, so.
Okay, so we are going to take into account uncertainty. We're going to look at ranks, but also relative treatment effects, which are measured with uncertainty. It will be a sort of there are multiple factors to take into account in the decision. So there's the ranking for efficacy and the ranking for safety, and also the cost of the treatment. And it might be that the top 3 ranked treatments for efficacy might actually be fairly similar. So we'll look at the relative treatment effects. If they're quite similar treatments, then maybe it will be safety or cost that determines the decision. So we are going to take into account uncertainty, but it's complicated. Sorry.
And just to add to that, I think the threshold approach is really important. Knowing that you've got 2 or 3 antivenoms that have got over a certain degree of efficacy with a safety profile that's good, I think is actually really useful for the way currently, or is a huge improvement over the way currently antivenoms are used. That would be game changing in itself for somebody to know I can give 1 or 2 or 3 of these antivenoms and know they're going to be efficacious, because at the moment that's just not the case.
Okay. Question over there? I think you had a hand up.
Hi. I read the papers that you've all been writing just about 2 days ago, so I'm new to this. So my main question is about the relative utility of family-wise error rates and pair-wise error rates. Probably, the reason I'm probably thinking about that is the original hypothesis tests and 2-arm studies are based on stone-cold refutationist logic, as championed by Karl Popper in the 20th century and invented by David Hume in the 18th century. So, bear in mind the first bit of my question, are any of you slightly concerned about maybe deviating away from stone-cold refutationist logic?
You want to? Okay, Becky, then Sarah, then
So, yes, we aren't looking at type 1 error rates. We do feel that the rankings are the most important thing, and we're not testing because there isn't a standard of care to test against. We are working, yes, with treatments which are generally already licensed, so we're not in an environment where we have to prove testing a type 1 error in order to use the treatment at all. So, yeah, I don't think we are very concerned about type 1 error rates, but I see your point, so we do have to defend against that.
No, I think it's a really critical point, and actually this was one of the biggest questions when we started thinking about these designs. How are you going to define what's good enough? Yeah, because I understand what you mean about the long history of statistics, but a lot of that was about saying, is something better than an existing treatment? And when you're in this scenario where, you know, these babies are pitching up at the units, you've got massive amounts of resistance, we're giving them drugs which, you know, have got neonatal licenses based on, and I'm not joking, data from one tiny baby 40 years ago, you know, that is the level of evidence. You know, what is good enough for a clinician? And what a clinician really wants to know is that I'm not giving something terrible to this tiny baby, and actually what I'm giving them has got a reasonable level of efficacy and potentially other advantages. And so that's where this concept of information gained, I think, really came from. This isn't drug licensing. These are all things that are out there and they are being used, and we just don't know whether we're making good decisions on a daily basis. So we have very little information. And so it is more of a Bayesian approach. You know, it is about saying what's the probability that these are in the top 3. But it's very focused on this idea that actually one of the key things about statistics is to decide, you know, what information we've got, going back to the point of uncertainty, how confident are we, and that's how the sample size is generated. That would be my take.
Thank you. Okay, go on.
Perhaps I could sort of kind of come in slightly. I mean, you know, it's not as if what we're doing now is good either. So, you know, for all new antibiotic trials that have been conducted in the last 25 years, but particularly against multi-drug resistant infections, there is absolutely no evidence that efficacy that is better than the currently available very old drugs from the 1980s. So there are many studies have done sort of all that's been done is there's no comparative data, no comparative studies, and the regulatory trials have been small, very poorly designed, sort of very, you know, and haven't provided any of the clinical evidence that we want. So, you know, regulatory sort of trials in antibiotics have completely failed to provide the information that is required for any sort of technology appraisal to provide any understanding of how countries can use what they want. So it's there's sort of, you know, it is a recognition of the fact that actually, unless we have some sort of kind of ranking of what is a potentially reasonable option, given that country's resistance problems, for example, or you know, then at the moment, the decisions that are being made by clinicians, as you can see by the 200 standard cares, are effectively random.
Thank you. Helen, if you could take the mic up there, but I'll do one online while you're doing that. So this one I think is for you, Becky. How would misspecification of the pattern for a patient, maybe the clinician getting the pattern wrong, for example, affect estimation of the treatment effect? Yeah. So the impact of the pattern being wrongly stated.
Yes, okay, so this is, so it might be, so might that be that the But yes, there's a treatment which is contraindicated, which is in the pattern.
I think that's the way round.
Yes.
Yeah,
I feel like the implications of that are more clinical rather than statistical, because it would mean the patient is being randomised between treatments where there was one treatment that wasn't appropriate for them. So that could have very bad clinical consequences. I don't feel that that's such a statistical problem.
I'm happy to have a go. I mean, I think the problem is it happens in standard trials too. I'm afraid and you can argue it happens less in placebo-controlled trials, although I've also had it happen in placebo-controlled trials where it's only after randomisation that the clinician goes, "Ooh, actually, yeah, maybe this isn't a good option for this patient." So, I mean, I think, if it's 1 in 3,000, the implications are very small. I mean, I do think, I presented the practical design as an individual patient choice. And I think because of the challenges with CPE infections, that is still true. But it is one reason why both David and Mike have talked about, rather than the randomisation list being an individual patient, sites will say these type of babies, these type of snake bite, so patients presenting coagulopathy in a site would get randomised between these 3 things. Babies who present, early onset sepsis, born outside hospital, will get randomised between these 3 things. And I think that's one way to practically mitigate against having much larger percentages of people ending up being randomised to things they should never get and then dropping out of the trial, which I think is a problem which, as you say, Becky, happens in the trials.
Okay, thank you. Question at the back.
Yeah, great. Michael Marks, London School. Lovely talk. Becky, in the simulations you presented, I just wondered to what extent they are dependent on assumptions you make about the proportions of patients who are eligible for those things. And for example, if you have a scenario like in David's where maybe 5% of the patients can actually only get drug D, whereas 90% can get drug A. So when you do your simulations, do you assume even distribution, within the populations? How do you handle those differential distributions of people being eligible for each of the different treatments?
Yes, that's an important feature of the simulations, and we did look at the sensitivity, and we looked at various scenarios. And yes, if you have sparser information, then you need a bigger sample size. So if there were if you have lots of treatment patterns where people can take most of the treatments, you have a lot of information, the sample size is smaller. If most people can only take, say, 2 treatments, you have less information, you would need a bigger sample size. So we did look at various different scenarios and different fractions of people having different proportions of patients when choosing both sets of sample sizes for both trials.
Can I just add, I do actually think that's a more honest approach than what gets done for most trials. Because most trials, you have one sample size calculation, you kind of assume that's fine and you go with it. And actually, particularly non-inferiority trials, as we were talking about earlier, if you get your control group event rate wrong, you can be in a lot of trouble. Whereas what we've done with these designs so far is really run a lot of simulations. I mean, it is a lot of work. But I think it gives you confidence that across a range of things that you might see, you're still going to gain enough information to make the trial worth running.
So there was a question online, and the laptop's just locked itself. My memory is, if you would please, Barbara, my memory is the question was for Becky. What's the implication of for practical use of these results? Do you have to get the personal randomisation list, as it were, recreated for a new patient who needs to be treated? And how would you do that? I hope I've got the answer. Yes, thank you.
I want to see you be a statistician, right?
Sorry, for the best randomisation list to be recreated?
Yeah, I'm sorry, Bob, I'm logged in. Using it, you'd presumably clinically have to guess the randomisation list the patient would have had they been in the trial, in order to work out which treatments to choose between. Is that right?
Okay, so I would think it would be...
You get the ranking of everything, yeah. I think it's similar to the question that you raised. So, I mean, the point is you rank everything just like you would in a network meta-analysis, yeah? Most people here will have seen a network and meta-analysis. you rank everything, but if then only the things in that ranking of 10 things that are relevant for your patient are number 2, number 6 and number 9, then the ranking is obviously 2 is better than 6 is better than 9. And I think the point about uncertainty is then critical, because obviously 2 may be very close to 6 or it may be very far away and so on and so forth. But the concept is exactly the same with the network meta-analysis. You use direct and indirect evidence to rank everything you've got, and then you take out of those ranks. I'm not sure whether the question is actually about heterogeneity in your point about indirect evidence. I think it may be trying to approach
I think this one's about practical use of the results.
Okay.
Yeah, so the decision process would be the same as it was in the trial to decide the treatment.
Yes, and I think it's sort of kind of I think there's two components to that. One is the sort of kind of the implementation of the study results, obviously, in terms of the sort of at a country level or at individual unit level, because actually, as Sarah was talking about and as David was discussing, these are then, in the end, they're both these studies are designed to inform how that might actually influence clinical decision making at a unit level, where they only have a certain number of drugs that they can afford or is available to them. Lots of issues with stock outs or availability of particular medicines. But then also how a sort of kind of country might decide to implement that, and the issues associated with potentially buying slightly more expensive drugs, still off-patent generic antibiotics, but certainly cheaper than gentamicin. But certainly more expensive than gentamicin. But actually, how and then the impact of that on neonatal mortality. So that I think is the principle of this is that to try and how this would inform a technology appraisal, so how this would then inform the sort of the TA process at the country level. I have to say that to some extent, because it's taking a network meta-analysis approach, and that almost in a way is more structured and hopefully will be better. Actually, that's not what scares me. Actually, what scares me is taking this to a WHO guidelines committee, which is predominantly going to be based on a large number of individuals who are very used to a standard trial design, and therefore their interpretation of what this might mean for a sort of WHO guideline, I think, is going to be the more sort of challenging in the spectrum. So that the ethics and regulatory at the beginning, the design, running the study through the middle, but then sort of kind of at the country level, I think you can almost see with good TA processes, that's okay. Thailand, for example, is a very good example. Someone will just take this data and go, yep, I can see how we can do this. But actually, I think other international guidelines committees may struggle with that, because that's the point going back to Becky. Because if you're doing this with generic older off-patent drugs, then in fact that's sort of kind of burnt. The struggle I think that actually we may well come into in the future, of course, is how you then take in new drugs where you're then trying to actually get regulatory level data within the sort of adaptive platform design, such that you can actually conduct these public health trials and then work out what a new drug has it had benefit of, or does it just add cost, and how we can make that level of data. Sarah,
No, I don't want to, Mike, actually, but I think what I would like to say is I think guidelines is very challenging, because let's face it, WHO is still recommending AMP plus Gent.
Yes.
So, I think there's lots of work to do. We didn't claim we had all the answers solved, but I think what we do claim is that sometimes the best thing to do is to actually have a good idea and get on with it, and we'll work it out.
So I think we should just take one last question, Helen, one right up at the back there again, please.
Thank you. Do you collect detailed data on the reasons why people were given a particular randomisation list? So, for example, if you have a patient in front of you and you, maybe their renal function is a bit borderline, and you might be able to give the drug, but you might not be, or their minimum inhibitory concentration is a bit borderline. Presumably, it's quite helpful to know, if one treatment does seem to be superior, whether or not patients like your patient would have been randomised to that option.
Mike?
Yes, it's a simple answer to that. In terms of, I mean, clearly, it is an issue, because obviously these, as with David, these trials are, this is very much a public health focused trial. So a number of the more sophisticated sort of kind of test availability that might be identical, so liver function test not available, already routinely available in so many settings. So I think it is, yes, but I think is probably the answer to that question. Yeah.
Okay, I think at that. Oh, is there one more? There was another one. Okay. Go on, just a quick one. It's going to take 5 minutes to get the mic to you, isn't it?
Who was it?
Oh, I'm sorry.
Okay.
Yeah.
Thank you.
Thank you. I was wondering, from a sort of clinical pragmatic point of view, in these situations like the neonatal sepsis example, where we sort of have equipoise, what you have currently is random care based on who the admitting clinician is or potentially which hospital you're at. And what my question was is, do you have a sense of when it's worth doing these trials prospectively versus looking at the existing data across what is random treatment and doing, say, network meta-analysis of existing historical data?
I mean, briefly, I mean, obviously, so we've, I think the neo-ops, we actually did do a large prospective observational cohort study, so we didn't go straight into the trial, in part because the data out there was so poor that it couldn't define a population group that actually is we know is of a high mortality, or we know is at risk of adverse outcome, and that's led to the sort of, the neonatal sepsis model sort of scoring system to try and derive that. I think the problem is that all of the other data sets that we've been trying to identify, have so much limited data, so much missing data, so many problems with data, so many biases in the way that systematic biases in the way the data has been conducted. For a different project, we've been trying to identify data sets that will actually identify whether you're on the right antibiotic related to your microbiology result, concordance, discordance, using the Gram data studies. The number of studies that actually contain globally from all the Gram data, bear in mind that was data from many millions and millions of already collected data. So the number of studies that actually provide all the data you require, which is the clinical information, all of the mortality outcome, all the microbiology, all the drug treatment, all the switching, all the forensic content, is 5. And they're not very good either. And that's it for AMR in its totality. So I think, prospective big large trials is the only way to get data.
So I think we should pull the session to a close there. Thank you very much. I want to hand over to Max to just sum up. But first of all, can we all thank all of the speakers very much?
So this won't take long. It's been a long day. So the aim of today was to show you how far we can move with innovation. When I started in clinical trials, I remember speaking to a number of clinicians, and they said to me, well, all the clinical trials methodology is sorted now. We've just got to do the trials. How much that has changed, even just in the last 10 years. And I'm sure there are more innovations and developments to come. And we shouldn't be frightened of innovating in this area because historically there hasn't been as much innovation in this area. We shouldn't be frightened of it. And there is a real capacity to do these things and an appetite to move things along more quickly. We've shown you some examples today, which I think, hopefully, you'll agree, will have a real impact on the field. And just before I move on to the final bit of my presentation, I'll make you 3 offers at the end of this. But before I do that, let's just thank Helen Smith, who's been central to arranging today. Thank you very much. She's not here. Oh, there she is. Okay, so Helen, you've been brilliant. And also, she's told me to tell you, downstairs is closed when you go out, so you can't go downstairs anymore because there's a party or something. And secondly, please leave your badges. In interest recycling, please leave your badges on the table as you go out. So I'll just finish by giving you 3 offers as we go. Firstly, all the questions that were posed that weren't responded to. There were a number on the Menti and we didn't get round to everybody. What we will do, if people have got the appetite for it, just send us in any further questions and we'll send you back written answers and we'll put them on an appropriate mechanism that people can access them. So feel free to send those in. And 2 further offers for you. Firstly, if you're interested in implementing or developing or talking about or implementing any of the designs that you've seen today, please do get in touch. And the final offer is an even bigger one, is if you've got a problem that's requiring a randomised trial of multiple interventions of whatever form, then do come and speak to us. Okay, shall we finish the day? Thank you very much all.